Hi folks, I'm Raif Derrazi and I am one of the co-chairs for the HOPE Collaboratory, which is one of 10 collaboratories that comprise the Martin Delaney Collaboratories funded by the NIH. So I'm here at AIDS 2024 in Munich at the conference, and several of the collaboratories have worked together to create this space called the Cure Canvas so that we can talk about HIV cure research. So I decided while we're here, why not interview some of the PIs or principal investigators of the other several collaboratories to talk about their work and how they're engaging with the community. So if you would please introduce yourself, tell us your name, the collaboratory you're with, and a little bit of your background expertise.
Okay, thank you very much. My name is Maria Salgado. I work at IRSI. I'm part of the HIV Collaboratory. My expertise is in HIV cure, and all my expertise is working with people with HIV with exceptional situations as the sessional controllers. I'm part of also of ISystem, the collaboratory of the transplantation, and now we are happy to be collaboratory with the Read HIV. Also for find HIV cure and another important cohort that is the Loviret.
Hi, thank you very much. I'm Marta Mazanella. I also work at IRSI. And right now I'm not member of any of the collaboratories, but I used to be from the Care Collaboratory from the beginning. Yeah, and I've been working like for the past 10 years in HIV reservoirs and cure, and specializing basically at different ages. So I was working with a pediatrician, pediatric HIV, like vertical transmission, and now I'm more focused in the HIV cure, basically in the perspective of age, how the age can affect the HIV reservoir, and to get like in the reactivation of the viral reservoir, and how this can impact like the strategies for HIV cure.
Okay, great. That's fantastic. Can you tell me specifically, RID, what is their primary strategy towards HIV cure?
Yeah, so actually Read HIV is quite unique in the way that we are researching HIV cure from the point of view of the innate immunity, more related with many omics. We are focusing in many omics. We have a cohort that is of people with HIV with very low reservoir, that they already have very low reservoir. Naturally, we are trying to find the ways that using metabolomics.
What is that?
Metabolomics is how in blood, how can the different metabolites, how different things that are coming from our food, our style of life, how can this affect to the HIV cure, actually, to the HIV reservoir, to our reservoirs in the body, and how can we eliminate this reservoir, then trying to find it a cure based in our results.
So it's about how environment, nutrition, things like that impacts
in the innate immunity, and how this finally the innate immunity impacts in the reservoir. Exactly. So the innate immunity is very important in the establishment of the reservoirs, because it's the first barrier that the virus is finding when it is getting in the body. So our hypothesis is different, as you say, environment. Metabolites or different conditions in the environment, how can this affect to your immune system, specifically more to the innate immune system, and how can this can be related to having lower reservoir. So like that, we can find different factors in the environment that can help to reduce the reservoir and in that way to trying to work through to an HIV cure.
Can you touch a little bit on what you are finding that has an impact?
So far, I mean, we have a cohort of around 1,000 people. From those, we already screened how the reservoirs are, and we found actually that in the last years, we saw an increased number of people with low reservoir. That means actually that different condition is not that much still in the environment, because we are trying to check different clinical factors. But what we found is the different clinical conditions. I mean, for example, initiating with the antiretrovirals earlier, For example, the introduction of the Innate, the integration inhibitors drugs. For example, all these kind of guidelines, new guidelines that were coming in the last year, have a great impact in the HIV reservoir. In the way that we see that actually, specifically from 2007 to now, there is a cure in the reduction of the HIV reservoir in the population that we are observing. So now we are getting these people with lower reservoir, people with higher reservoir, and comparing these innate immune factors. And we are starting to see some signals, immune signals, based in different immune markers that can be related with lower reservoir. So we are now, right now, getting more results, and hopefully soon we can get more results. But we are very characterizing very well these kind of people.
So you're able to measure specifically the size of the reservoir.
Exactly. Yeah.
Can you tell me where in the body the reservoir is?
Well, the body, the reservoir is mainly in the immune tissues, but mostly in the gut. It's like 60% of the reservoirs are in the gut. Also, around 30% of the reservoirs are in the lymph nodes, and less than 1% of the reservoir are in blood. But blood is our tool. At the end, the tool that we use mostly is the blood. So right now, the screen that we have is in blood because it's easiest for us. But in a previous study that we did, we checked for the people with lower reservoir. We check if this a correlation with the reservoirs in blood and in different tissues like gut and lymph nodes, and we observed a very good correlation. So the people that we saw that it was very low reservoir in blood, they have very low reservoirs also in the different tissues that we check.
So even though there's the least amount of reservoir in the blood, you said 1%?
Yeah.
About? Because it's proportionate to other locations in the body, you're still able to use blood. It's a good indicator.
Exactly. Okay. So in the clinics, usually we check CD4s and viral load, right? And that's what people is used to know. Even with undetectable viral load, if we get the blood from this person with undetectable viral load and we go to the lab and we check the reservoir, we are detecting very good amounts, higher amounts of the HIV reservoir in the laboratory. So we see something that the clinician usually don't see, that is the reservoir. And that reservoir that is hidden is the cause why it's needed to have the long life therapy, right? Because if the therapy is stopped, we still see the reservoir and the reservoir start to replicate. But in blood, we are able to see very well.
And so how does your research in innate immunity help towards achieving a cure?
So the idea is to find different markers, right? So if we have different environmental markers, there is something that we can do at least to reduce the reservoir. So reducing the reservoir is the first step. Then we try to eliminate completely the reservoir, right? And even we need to give more power to the immune system later on. So even if we see some markers that are telling us this is reducing the reservoir, but also these markers are related with the immune system, we can, in a double way, to improve then the immune system to block this viral rebound in case the therapy is stopped.
Okay, fantastic. And then I'll ask you a little bit about you're working with, tell me again, your research, it's not pediatrics anymore.
It's not pediatrics anymore, but we are focusing at different age ranges. So because as we age, that it's something that we do all like naturally.
Unfortunately.
Unfortunately, yeah, of course. Our metabolism, like the way we process the energy is different. And we think that there is a problem not a problem, but there is different ways not to activate the metabolism, and that impacts in the HIV persistent and the reactivation of the virus. So maybe the strategies for HIV eradication, they are not the same from younger population than from the older. And that comes from studies that I previously did in my postdoctoral studies, that with a similar level of HIV DNA that is measured in blood, that is usually what we measure like from peripheral blood, like children reactivate much less the HIV reservoir, like getting these infectious particles, like this reservoir that we want to eradicate, than the older population. So that's why we were focusing like in these age differences, that it's a little bit similar, like in the same way that she's studying the metabolism, the metabolites, I'm going deeper at different age ranges.
So with children, you're saying that the latent reservoir it reacts slower? Or
Yes,
reactivates
actually,
slower?
yeah, we were working with a Thailand population, like virtually infected, like children, and in there we were measuring, like we were able to measure HIV DNA, but we were not able to measure this inducible HIV, this one that is we need to eradicate. So, and that was really different from other cohorts, like from adults. Like with the similar level of HIV DNA, we were able to induce this HIV reservoir.
And so how does that inform a potential HIV cure?
So the things, HIV, like for the reactivation, it needs the metabolism of the cell. So maybe it's a new way to tackle and to reactivate this virus, or the other way around to make it like more silent.
Permanently silenced.
Yeah. So I think
So it could be either one.
It can be either one.
Okay.
So we are really at the initial stages. So I anticipate we don't have results yet, but that's our aim.
So just to be a little clear, she's saying that there's two angles of attack that are potential here. One is to activate the latent reservoir, activate all of it, 100% of it, and then as we know, our ART will kill all the active HIV, and then you're cured. Or we just completely silence it so that it's never replicating. We can stop ART, and it's latent reservoir, and it's permanently silenced and can never reactivate. So that's
the two ways that we can try to tackle.
Cure.
Yeah. Yeah.
That's what we know of in the London patient and
Correctly.
the Berlin, and so
Yeah.
for each of you, this is a little personal question, but what was your inspiration, motivation specifically to focus on HIV cure research?
Well, I mean, there are many. I mean, from my beginning of my experience, I always found I started with what it was called long-term non-progressors at the beginning of my thesis, where it was these special people that the virus didn't progress.
Yeah.
And one time, one of these person came to me and say, "What's different to me? Why my friends? In the 2000s. It was my friends already died, and why I didn't, right?" So having that idea and that question and try to make people don't die. Now we have antiretrovirals, and it's much better, right? But at the beginning, it was the idea that like why some people can control the virus. How can we put this to everyone,
Was
right?
that in a healthcare setting?
It was, yeah. We were working in the lab, but with people that was not progressing. It was in that time. It was long-term no progressors. Later on, was elite controllers.
Yeah.
Part of half of my career was based off elite controller. Then I follow all the transplanted people from the eye system. So kind of these cases are always a hope that a cure is possible, a functional cure in the people that automatically control, like the controllers. I mean, they have very good immune system, or even like with intervention, like we happen with the transplantation, right? There is a hope.
Yeah.
There is people that we observe that there is a functional cure, and their bodies are able to control the virus. So for me, like the progression that I saw in my career with these special people is always giving me the idea that this cure is able to be cured, is possible.
Yeah.
And the idea is to try to find that cure for everyone.
And even more broadly, why HIV research in general?
Well, I think first, I mean, now I know that we have now antiretrovirals and life expectancy
But
is
when
quite
you
good.
were
But like HIV, like from the biological point of view, is an amazing research because you are mixing immunology, you are mixing virology, you are mixing metabolism. There are missing many things. Like for the learning point of view, it's an amazing school. It's really amazing school, and it's very good way to learn many aspects of the biology of the human being. So it's very interesting to see how a virus is creating all this environment and how the immune system cannot control, can be with some problems, and it's always fun because you learn every day, right? And finding a cure is amazing. It could be amazing also for everything that is around the HIV, right? Like there are many feelings around the HIV, so finding a cure, I think it could be very interesting for the people also to solve those problems with the feelings, right?
And for you, same question.
Yeah, it was a little bit the same. So when I took the microbiology course, and the virology was inside there, I thought that there was a really big problem, like with the epidemic, with no cure. No, like people, like they were starting the antiretroviral therapy, was working already, but there were no cures. So I really wanted like to help people and to try not to end the epidemic, right? And I think that's why how it started. At the beginning, I was not focused on the eradication or the cure, on the cure strategies, but I work with discordant patients, so those that they were taking the antiretroviral therapy, but they were not able to recover the CD4 counts. And like slowly, like they fused like the studies, and we were studying the HIV cure in this population specifically. And this is how I entered, and I found like that really interesting from the biological part of view, but also to trying to get to my initial like dream of trying to help the population. Yeah.
Thank you for sharing that. And then, so from a community aspect, how are you engaging with community?
Yeah, so probably because I work in the lab, so sometimes for us it's not that easy. But probably the project that I engage me more with the community is the iSystem project. I had the chance to work in the lab without knowing anyone from some of the cure patients right now, and then now I'm having the chance to know them, to talk to them, to know the other aspect of the story. And for me, it's something that actually is helping me a lot to understand many things around the HIV and the stigma behind that, and even how the cure can change also many things, a lot of hope for many people. So, and actually, inside of the collaboratories, I think is a very good chance for us also to know the community, to know how they think, because these kind of programs actually are including the community in very active way, and it's a way also that the researchers that are in the lab,
Yeah.
and sometimes we don't have that connection. I'm not clinician, so I don't have the connection sometimes.
You
We
get a
have
chance to engage with the community advisory board?
Yeah, I mean, we are engaged in the meetings. We are always in contact in the meetings, and we try everything that we do. We always in all the meetings every week. We talk with the community. We have always representative of the communities in the Read HIV, and it's a very good way that there is a two ways research, right? Like sometimes we get in the lab and that feedback is helping us a lot to also kind of modulate different things in our research.
So it's a little bit the same. So I think it's really important to have this connection, because in the lab we are focusing ID of the patient, of like it's not humanized, like we don't have the feeling, and I think it's really important for all the basic researchers to get this engagement, and this experience, to see outside of the petri dish, as we see. So I think it's one of the things that I appreciate the most, like to have this interaction, that I think it has coming up like more like recently, and I think in Spain we need to keep working on this aspect, because it's not getting where it's I think like in the US it's much more advanced. What is
I was going to say, because with the Martin Delaney Collaboratories, it's part of the structure, is that we have a community advisory board and there has to be community engagement. Is there any kind of formal structure there, or is it kind of on you to sort of?
It's more on us. So it's something that we want, but of course to do that also you need some money, some resources to be able to like put together all the people in the same room and everything. And I think it's something that it's missing in the Spanish research. I don't mean that we don't do it, but I think we should work more on
The US is fortunate to have
I think so, yeah.
a lot of funding.
Definitely. I mean, the structure of the Martin Delaney's, I think, is very unique, because it's one more partner of the research in Spain. I mean, we have the connection, for example, with Checkpoint in Barcelona, that is working also in some of the Rate HIV meetings, but we have the direct connection with them, but it's not like a structured programs in the funding that we get, like it happened with the Martin Delaney's. And I think this is really rich, at least for me, for myself personally, it's been very rich to have this interaction.
Okay, and then for the final question, in the next year, what are you hoping to learn more from, gain from your current research?
Well, I mean, in this project, actually, in the Martin Delaney project, we are right now running many experiments. So hopefully, we can have in the next year have really kind of a China tour of with these main factors that actually can check what is related to lower reservoir. I think to have that signature could be very interesting to know how can we separate, I mean, different factors that can be related with low reservoir. We are working in different projects, right? Probably you as well, right? So and science is running fast. We are still working with transplanted patients, so still there are some people with HIV that is there. They were transplanted, and probably they will stop treatment the next year. So probably we'll have more people in this party, this happy party, the people cured of HIV. And I'm waiting for getting any surprise also, because science is like this, always new surprise.
Well, hopefully we can follow up in a year and see.
So in my case, since we are starting now the project, we would like to know like a little bit more about the metabolism in different age ranges.
Yeah.
But at the same time, we are investigating also how the aging of people with HIV, because as you may know, like even though the life expectancy is the same, a lot of comorbidity starts to appear in at earlier ages. So we are studying at the same time this kind of like to see if there is an advancement of the immunological age, that like immunologically, if like people from with 30 years old are the same as like 40 or 50 of non-infected individuals.
So to clarify, there's a difference between chronological age, which is the age we measure in years, and then the age based on our
immunologic,
our immune system. So you might hear that sometimes. Oh, I'm 39 years old.
You look younger.
But maybe I have the immune system strength of a 20-year-old. You know, it doesn't have to match the chronological, and HIV can have an impact on whether that's older or younger, depending.
Yeah. So usually HIV tend to be older, because like the replication of HIV, especially during the first acute phase, it may damage the immune system.
Chronic inflammation.
With the chronic inflammation, a little bit more of immunosens. That's why sometimes, like with the response to vaccines, like to tetanus or hepatitis, there are like with there's less strong, like they are weaker, like you might need like more like injections or more like. So we are trying to study this kind of things, like if the immune system is more immunosenescent, like more aged, and see how we can revert that. That's
and
it.
how you can revert that.
Yes. So keep tuned.
Yes, very interesting. Thank you both so much for sharing some of your research, and
thank you
it's
to you.
inspiring, and I'm sure it's going to give my audience a lot to think about, and I'm sure they'll have lots of questions too, so maybe we can follow up at some point as well.
Thank you very much.
Thank you very much.
Until next time, cheers.